Light signals are generated once cells or particles are interrogated by the laser. The light signals are converted to equivalent electronic signals and then further processed and converted to a number. The larger the signal, the higher the number. The data is stored by the computer system in a flow cytometry standard format developed by the International Society for Advancement of Cytometry. When the computer saves data from the cytometer, it is saved as a list. The numerical values for each cell's measurements are saved. Data can be displayed in different plot types by plotting the numerical values. For example, in a dot plot, two parameters are plotted against each other. The FITC value for event 1 is 690, and the PE value is 875. The dot that represents event 1 is mapped on the plot at coordinates 690 on the FITC axis and 875 on the PE axis. The FITC value for event 2 is 39,271, and the PE value is 90,000. The dot that represents event 2 is mapped on the plot at coordinates 39,271 on the FITC axis and 90,000 on the PE axis. We can see that after many single events are graphed together, populations start to emerge. Data can be displayed with different scaling choices. Traditionally, flow cytometry data has been displayed using a log scale. However, data generated on a digital instrument and displayed on a plot with log scale might not be able to show all of the data optimally because some of it falls on or below the axes. A plot with bi-exponential scaling turned on displays events that fall on or below the axes. Bi-exponential scaling can also improve resolution between poorly resolved populations and is useful when analyzing complex data.
Display a single parameter against the number of events or counts. Events with higher fluorescence intensity appear to be on the right on the x-axis. Dot plot displays two parameters simultaneously. Each dot represents one or more events, cells, or particles. Dots appear further to the right on the x-axis or higher on the y-axis as signal intensity increases. Density plot displays two parameters simultaneously, provides a third dimension using color to indicate the event count. Similar to contour on a contour plot, dot for coordinates with the same events count are given the same color. Dots appear further to the right on the x-axis or higher on the y-axis as the signal intensity increases. Contour plots displays two parameters simultaneously. Contour lines provide a third dimension by joining x and y axis coordinates with similar events count, similar to topographic maps. And not to leave out from the dot plot,
A gate is a numerical or graphical boundary that can be used to define characteristics to include for further analysis. Gates are used to identify subsets of data or populations. Populations defined by gates can be used to generate statistics and limit the number of events collected or saved. This example illustrates a human blood sample containing a mixed population of cells. To restrict analysis to the lymphocytes only, a gate is set on the FSC versus SSC plot. Then, the plot can be changed to display the fluorescence properties of only the lymphocytes.
Quadrant gates or markers divide a dot density or contour plot into 4 separate populations. Polygon. A polygon gate specifies a boundary around a population on a dot density or contour plot. Autogate. An autogate is automatically drawn around a cluster or range of events on a dot density contour or histogram plot. Once drawn, an autogate shape and size remain constant when new data is displayed in the plot. Snap to gate. A snap to gate is automatically drawn around a cluster or a range of events on a dot density contour or histogram plot. Snap to gate adjusts their shape and size when new data is displayed in the plot. Snap to gate are useful for analyzing sample stain with the same reagent combination, such as for data batches analysis.
Different colors can be used to display gated events. Different colors can be used to display gated events. This allows you to visually compare subpopulations that appear in different plots. For example, the blue-colored events in the lymphocytes gate on the left can also be seen in the plot on the right. You can see that the lymphocyte population in the plot on the right is split into several subpopulations based on fluorescence.
Data display. Data displays one or more of the many plot types available in the software application you are using. Two, create gates. Create gates to identify subset population of interest and assign colors to better visualize the different subpopulations. Three, display statistics. Display statistics to view a distribution of events. Analyze data. Review the display data, gates, and statistics and make observations and conclusions about what the achieved results mean in relation to other known experimental conditions.
One way to analyze data is to determine the percentage of cells or particles that are positive for specific markers by setting gates, such as histogram or quadrant gates, while viewing data from a negative control. In this example, histogram gates indicate the areas of negativity and positivity. The histogram on the right shows that some cells in the test sample are positive for the PE-Cy7 marker. To determine what percentage is positive, you can view the percent parent statistic for the positive marker. In this case, 66.7% of cells are positive for the PE-Cy7 marker. Another way to analyze data is to study a specific population and the subsets of that population. In this example, a gate was created to identify lymphocytes in the FSC versus SSC plot. In a plot displaying only lymphocyte events, quadrant gates were created to identify subsets. The statistics displayed show the percentages of the subset populations as a percentage of the parent lymphocyte population and also as a percentage of total cells. To determine the percentage of lymphocytes that are CD3 and CD4 positive, you would view the percent parent statistic, 39.3%, for the CD3 positive, CD4 positive population. The analysis methods illustrated so far show percentage estimates of populations based on their staining for specific markers. To compare fluorescence intensities and measure the degree of positivity, compare the geometric means or medians of the control data versus the test sample data. If the test sample data statistic is greater than that of the control data by a limit set by the user, it would be considered positive. The greater the difference between the two, the more molecules are expressed per cell and the more positive or brighter the population. In this example, let's say the medians of the control and test samples are 2, 5, and 20 respectively. If the researcher set the level of positivity at 6, then test sample 1 would be considered negative, and test sample 2 would be considered positive. Reflect on what you have learned in this module. How is data saved and displayed? What is the purpose of gating? What are examples of data analysis methods? Now that you have completed this module, you should be able to describe how data is saved and displayed, explain the purpose of gating, state examples of data analysis methods.